Good morning to all. I welcome you to the webinar series 2020, which is organized by Department of Zoology, St. Joseph's College, Devagiri, Calicut. Before starting the session, I would like to request the participants to follow the following instructions. Please mute your audio and video during the entire session. Queries and concerns may be posted in the comment box. A separate interactive session will be arranged at the end of the presentation, during which you could interact with the presenter. After completion of the program, carefully fill up the feedback form. The e-certificates will be distributed based upon this information which is entered in the feedback form. So please be careful with their spelling, capital letters, etc. Okay, now we move on to the main session. As you know, this session is on the role of Orexin in sleep and neurological diseases, and this is being organized by the Department of Zoology, St. Joseph's College, Devagiri. Now, I invite Dr. Bobby Jose, Head, Department of Zoology, St. Joseph's College, Devagiri, to introduce today's speaker.
Good morning. It is my pleasure to welcome everyone to the international webinar organized by PG and Research Department of Zoology, St. Joseph's College, Devagiri. I extend a warm welcome to Dr. Dr.Thomas, Principal, St. Joseph's College, Devagiri. He is a member of the Zoology Department also. I also welcome faculty members, research scholars, and students from Devagiri College and various institutions in India and abroad. Today, we are lucky to have an eminent personality, Dr. Thomas Thannickal, from the School of Medicine, University of California, Los Angeles, USA, who is well versed in the field of brain research. He is an eminent researcher in the area of neurodegenerative diseases, especially narcolepsy. Dr. Thannickal, along with his colleagues, discovered the role of hypocretin, is also known as Orexin, cellulose in human narcolepsy, and eventually opened up a novel area of research. Presently, his research is focusing on the roles of hypocretin in Parkinson's research and opioid addiction. He published numerous research articles in leading journals, including Science, Brain, Neuron, etc. During his 24 years of research experience, Dr. Thomas worked in association with several well-reputed national and international institutes. He has served as research officer in All India Institute of Medical Sciences, New Delhi, and has published a single author essay in the journal Science as part of Eppendorf and Science Awards. His contributions has made significant impact in neuroscience research and received worldwide attention. During the research career, he received University of California, Los Angeles Chancellor's Award for Postdoctoral Research Excellence in 2001, and Sleep Research Society's Young Investigator Award again in 2001. He was also selected as a finalist for the Frank Brown Berry Prize by US Medicine and was a runner-up for the Eppendorf and Science Prize for Neurobiology from the American Association for the Advancement of Science in 2002 and American Academy of Neurobiology Sleep Science Award in 2003. I have great pleasure to inform you all that Dr. Thomas was my classmate during my MSc at SB College, Changanasheri. I cordially welcome Dr. Thomas to this webinar. I hope you have an engaging and enriching session ahead. Thank you all.
Before inviting our speaker, I would like to invite our principal, Dr. Thomas, to add a few words in this seminar.
Good morning to everybody. And it's a very happy occasion for me to be part of this program because it's my parent department, it's my area of zoology, and more than that, I was taking classes for the same topic the sleep and the sleep disorder, sorry, the sleep waking homeostasis for a long period of time. So first of all, I am very much thankful to Dr. Thomas Thannickal for sparing his valuable time. University professor from California, a person with such a reputation of long list of awards. I see he's having a sleep science award. Then there is the award from another award, which I know as the American Sleep Research Society's award is there. Then Frank Brown Berry Prize, US Federal Award. See, getting an award in a US society, it is not like what we see in other parts of the world. It's a reputation. And having publications in journal like single author publication, Science and all, speaks about him. And you might have been watching some 10 to 15 minutes. You got an opportunity to interact with such a well-known scientist, established person. And from his biodata, it was an occasion for all of us to understand that. He was like all of us, BSc Zoology, MSc Zoology, but he has his own plans going all the way to AMS and finding opportunities, understanding what to do, and then from there venturing out. That is the best part of it. So there's a saying like, one race gathers honey. The people who go out, difficulties will be there. So Dr. Thomas, I take you as a best example for all this, because for the past 20 years, I have been hearing about you from Dr. Bobby Joss, my head of the department, my colleague. He often says about you. Unfortunately, we could not bring you, but now thanks to Corona, we got an opportunity where it is an online presence, you are here. Yeah. So then my colleagues in the department, Dr. Vinesh, Dr. Jisha Jacob, Dr. Ashwadi, and Dr. Arnaksharan, Joy sir, they are all very much behind this program. And it is quite nice to see acting as a team and arranging a very good program, meaningful program, because this is a subject-oriented program is useful for BSc students and MSc students also. For BSc students, in what way you might ask me, because see, you will be studying the basics of physiology and all. That's what Dr. Thomas was telling. Sometimes you'll be wondering what's the point studying all these, what is the idea of studying brain and physiology and all dry subjects like that. See, those are the areas. Now, then I was seeing in his biodata that is Parkinson's disease and Alzheimer's disease is working with. Since I was taking that particular topic for some time, that is the genetic base of Parkinson's and all, there are most of them, what I saw, there is currently active research is going in countries like Western countries. That means there are opportunities. So the people here know you can be in contact with Dr. Thomas. Not for each and everything, because he's a busy person. He'll be deeply involved in his work and all. But meaningful, see, when you have a good biodata show up with good grades, good publication, or some future plan, people like all the youngsters who gathered here, they should make use of such opportunities and aim for, or they will be able to guide you what is happening, what is the need. The earlier days of just passing UGC, CSR and all, getting a stipend, no, that's all over. Now that's what happening now also. Opportunities are for people who are daring to work beyond that. It doesn't end with your CSR UGC alone. After that, get into a good institute, good publications. And Dr. Thomas says he says one science publication that speaks about him. His biodata says everything. So that's what I say, he's a role model. And we are very lucky to have him here. And he's a colleague of Dr. Bobby Joss. And I sincerely wish that all of you will make use of it. And a special thanks to Dr. Thomas once again for, now it's award time, might be around 10 o'clock, and I know it is your selection results and all. You must understand that his 1 hour or more than 1 hour, he is very much willing to share for his 100 people. So thank you once again. Then there are people from Dr. Pannachan I am seeing here. He's a senior colleague of mine. Yeah, he was with St. Thomas and all. Yeah, retired 5 years ago. But I think Dr. Pannachan has joined to this. This was the interest of the people who are interested in it. So this kind of meetings, webinars and all, enables people to come together, join together, and to see what is happening, and then to plan out. It is not simply watching it all and going home. Go home and plan what to do next. And I don't want to continue further. So once again, I thank Dr. Thomas, Chacko, Thannickal for accepting our invitation and willingness to be a part of this program. Thank you.
Now I would like to invite Dr. Thomas Chacko Thannickal to present the session.
Dr. Thomas, your mic is off. Please switch on. Your mic is off.
Hello, sir.
Yeah. Dr. Thomas, can you hear it?
No, we are unable to hear Dr. Thomas.
Yeah, because his mic is off. Yeah, now it is okay.
Hello?
It's
Hello?
audible. Yeah, we can hear Dr. Thomas. Please carry on. Yeah.
So now, can you see?
Yeah, now everything clear. Your screen is clear. We can hear you. Please carry on.
Okay. So first of all, I would thank you, Dr. Thomas, and head of the department, Bobby Jones, and the program coordinator, Jisha Jacob, for giving me an opportunity to share my work. So for last 20 years, I am just focusing on one neurotransmitter, which is called Orexin. So the term may be new for you because this is the neuropeptide discovered in 1998. So this neuropeptide, there is another name called hypocretin. So why there is two names? One is orexin and one is hypocretin. So first I will explain what is orexin, the word origin. So orexin, it is the Greek word orexis, which means appetite. So there was in 98 two groups, one from California, one from Texas, looking for the hypothalamic neuropeptide, and they are making a hypothalamic peptide library based on the different functions, biological functions. So both group have same identification, but they named differently. So one group named orexin because it, from the Greek root, the role of orexin in a feeding. So the another group called hypocretin, that means it has the location, it is in the hypothalamus, and it has the weak resemblance with the secretin hormone. So they call it as a hypocretin. So the name orexin will be most of the time I am using. Sometimes there will be overlapping with the hypocretin. So I am working in the institute, University of California, Los Angeles. And we have a brain research institute. It is a part of the UCLA School of Medicine. And this institute has more than, for brain research, more than 400 faculty and from 30 departments. So for studying any of the thing, it is now, it is a very collaborative effort. And even one lab, it may not be able to answer all the questions. And because high impact publication means you have to approach multi methods and you have to answer all of the questions possible. So now it is a common practice that the interdepartmental, interdisciplinary group work. So when you see a publication in like Nature, Science, or Cell, there will be more than 20 authors, and you can see that there will be multiple countries, multiple universities. So it is a very common practice, and all of the instrumentation, we have a core facility. Even now we have the live imaging, and even we have the departments developed electrophysiology. Electrophysiology now modified into based on the optics. We can precisely determine the physiological properties. So now there is a deep focus on. So neuron is only one component of the nervous system. So there is. So another group is a neuroglia. So which consists of microglia, astrocytes, oligodendrocytes. So when we talk about these group of cells, it comes with a nearly 1:20 ratio. It is not exactly calculated, but people are getting interested with studying these cells because most of the neuronal function is regulated by neuroglia cells. So this is the summary of the work I am going to present. So in 2000, we have a major discovery with Orexin, that is the human narcolepsy. So that is until that, because I already told that this is discovered in 98. So there are major developments, and we have a big tight race with each and every publication. We have competition from Stanford, we have a competition from Johns Hopkins, We have competition from the European Union, Netherlands Brain Research Institute, and Yale University because they are highly aggressive. And most of the researchers in this sleep field now, MD, PhD, because now there is a maybe within last 10 to 20 years, there is a high clinical application for sleep disorders. So with the 2000 finding, there is a major landscape change with the narcolepsy concept. Previously, it is considered as okay, it has a similar symptoms of seizure, epilepsy. So in olden days, it is considered as okay, the patient has something, a mental problem. Okay, it's a psychotic. So it has all the psychological problems. So then, by working for narcolepsy, I have something with the follow-up with the Parkinson. Why Parkinson disease coming into the role of Orexin? Because Parkinson disease has most of the narcoleptic people's symptom except cataplexy. Then, while working for Parkinson, we have something unusual observation that one control subject, which has 50% more hypokinin cells. So we never looked. That is in 2007. We sidelined that subject, and after that, we followed up that patient, and that patient was 20 years of heroin addict. So that triggered for the discovery of a new neurotransmitter called, or the same molecule Orexin, which has a major role in the addiction, especially opioid addiction. So you can see that the importance of this work into our humanity. You see, narcolepsy, it is the very small percentage, 3 million, that's the total of world population. And you can see that Parkinson, it is the world population is 10 million. And you can see that the addiction problem, you can see that even if you look each and every news, even if I sometimes observe the Malayalam news, you can see that the community, the society is flooded with opioids, even addictive compounds. And that they are using, it is maybe 10 times, 100 times more addictive properties. That's the synthetic one. It is flooding in our society. In 2018, in America, the national emergency declared with Trump administration because there is nearly 500,000 people died. These are young people overdose. So you can understand how significant this research works for the human as a human society. So this is the two group. They discovered the hypocretin. This also the accidental because these are something with they have a big plan. They looked for the hypothalamus, and based on the new technologies, they are screening for the hypothalamic neuropeptides. The hypothalamus is the most important part in our human brain. When you because I have a lot of human brain, because my all the studies working on a human brain, and we have a national brain bank consortium, and for my study we collect most of the, usually the brain bank slices, okay, and there are the preservative is there are different methods, because sometimes it for the biochemical analysis it is -80 going to directly from the patient head to the -80. So some brain, if we have a special request, and it is a formalin fixed because we are using the pathology, and so we will get half of the brain which is fixed in formalin, and that we need because Usually the pathological analysis involves very small pieces, and they will slice into 1 or 2 slide 40 micron, and they will make a note. That is for the pathology. That is the mandatory here. And even any death, you have to report the government, and if it is not in the hospital, they have autopsy. So this is the summary telling that, okay, this I just scanned the publication related to this specific topic. It is discovered in 98, and until 2020 October, there is almost 5,626 publications. So this is very encouraging for this neurotransmitter, and it has a lot of groups studying focusing on this. And you can see that, okay, from within this publication, my work cited almost 3,225 publications. You can see that, okay, how it is almost 56% of this field publication. It is already following up based on my publication. There is the follow-up work going on. So now this neurotransmitter, it has a great potential for human health, especially the neurological diseases. So in the follow-up, I will be talking about what I did. It's a Parkinson and narcolepsy opioid. So there are so many neurological disorders which has reported that there is the role in the disease progression. This neurotransmitter has major role. So why this a single molecule? It has this many implication in our body, how it is happening. So in the later slides, I will explain why this is the important one. So before going to the details of the study, I would like to explain what is orexin. It's we call orexin family. So this in the body, it is derived from preorexin, and this preorexin is divided into 2 molecules, orexin A and orexin B. So, and it has for acting any neurotransmitter in our system, we need receptors. So this neurotransmitter has 2 receptors, orexin 1 receptor and orexin 2 receptor. So this is entirely located in the hypothalamus. You can see that, okay, this it is this area. This is around the 3rd ventricle. So hypothalamus is the very important area in the brain, and these are some of the cell samples given in the fluorescent. So this is just a slide showing the hypokretin and melanin concentrating hormone neurons, which is distributed in the rat hypothalamus. So this is, you can see that with this is the fluorescent, and you can see that this is the red, it is the orexin, and this is the MCH. So why I focusing on these 2 means because when we explain the orexin, we have to substantiate that this MCH neuron, it is intermingled with hypokretin, and this MCH neuron, it has also a major role in the sleep. So this is the diagrammatic representation. So you can see that the preorexin, and it has divided into orexin A and orexin B, and it has the 2 G protein, orexin receptor 1 and orexin receptor 2. So this is just summarizing maybe a half a dozen functions of orexin. So initially, they found, the discoverers found it is in food intake, that it is increasing the appetite. That is why they are called orexin, and it has an arousal. So the arousal state that triggered attention of narcolepsy researchers, because the narcolepsy is a REM sleep disorder. So basically, they are not able to keep up the arousal stage. So the follow-up study, it is conducted by Stanford, because in America, there is only one lab, it's Stanford, which has the narcoleptic model. It is a narcoleptic dog, which there was no animal model during that time. It is in 1998. So the Stanford group actively looking into, is there any role of this orexin in the cause of narcolepsy? So before going to narcolepsy, we have to understand what is sleep. So these are a hypnogram showing the sleep architecture. So sleep is a circadian function. We know that okay, sleep, it is a good. So usually in a person's life, 1/3 of time is spending in sleep. So what is the function of sleep? Then what is the role of in our health? It is very important to understand the physiological or neurochemical and electrophysiological functions of sleep, and we have to understand the mechanism. So this graph, just summarizing, it gives a brief summary, because the sleep architecture is very complex, because there are dozens of neurotransmitters and there are multiple location centers are involved. So even each stage, the stage 1, stage 2, stage 3, that is the non-REM sleep and REM sleep. So basically the narcolepsy is a sleep disorder of REM sleep. So sleep architecture basically involved in the organization of the sleep, and there are two types: non-REM sleep and REM sleep. So the non-REM sleep are stages, 4 stages, and REM sleep are the deep stage, and it lasts for 120, maximum 120 minutes. So what are the differences? How the stages are differentiated? Because it is quantified by polygraph, and you can see that these are the polygraph recording, and basically you can see the difference. So, and we have to record different physiological parameters. So that I just briefly few of them. So you can see that the active wake, then non-REM stage 1, how the physiological properties are changing, and even the electrical pulses are changing, and with the slow wave sleep and the REM sleep, which is a deep sleep, even the brain getting calm and quiet. So when we sleep, the brain is not sleeping. It is doing their work, backup work. So what is the role of sleep? Do we need sleep? So there are so many questions. So how we will define sleep? So when we take, okay, now the most model used Drosophila, and when we come to the different rat, mice, even a monkey, dog, human. So in our lab, recently conducted a study in South Africa with tribes. What we call is the sleep demand in industrialized nation and pre-industrialized situation. So our normal concept is we have to sleep for 8 hours. So even in America, even all the agencies are recommending 8 hours sleep. So we have conducted a study in a South African pre-industrialized population, and they were tribes. So we found that because we have used the instrument called ActiWatch, so it is not giving the complete data of a polygraph, but it gives 90% resemblance data, and they are purely depending on the nature, and their average sleep time is only 5.5 hours, and they don't have any significant health problem. Then why we need 8 hours in the how we need? So we cannot completely replicate the pre-industrialized lifestyle, because here there are a lot of environmental factors and lot of things are influencing. So this is I just telling that, okay, even now there is a strong concept that, okay, 8 hours. So even now there are population with a 5.5 hours average wake time, and also in the existing. So this is giving an overall idea how the major locations of the neural pathways of hypocretin neurons and their receptors. So you can see that the major arousal centers, it is going in the pathways. So it's going to the nucleus accumbens, it is going into tumoral mammary nucleus, and it is going to VTA, it is a dorsal raphe, and the brainstem locus ceruleus. And this figure, it is showing how the orexin molecule function as a wakefulness molecule. So from the orexin, it already putting pressure to get these brainstem nucleuses, and it controls the action of this other nucleus also. So when there is a shortage of this orexin, the person cannot be awake properly. So that is one of the sleep disorder. So these are the general, some of the patterns of sleep-wake disorder. And I am just focused only on the narcolepsy. There are insomnia. Insomnia is the most common in anywhere, especially in developed nations. And RLS, restless syndrome, non-REM sleep, REM sleep, hypersomnia, circadian rhythm. So what I am talking about, I just focused only a very small topic in this field. You can see that the sleep, it is not a simple thing. It is a very complex. When you come to the nervous system, if it is the most complicated, we to study the sleep parameters. So that is the one of the disease. It is called narcolepsy. That is my focus. So how the orexin molecule, which is linked to narcolepsy. So the same group who discovered orexin, and also Stanford group, they have a collaborative study, and they published in 2 publications. So they generated orexin knockout mice, because that means they are taking out the orexin from the brain. So there is no orexin available. And they also studied the narcolepsy dog, and they came out that when you remove orexin neurotransmitter from the body, you will get narcoleptic symptoms. So then that's the mice. Then what is the status in a dog? So basically, they studied the gene mutation. So they have not found any abnormality in the hypocortical neurons in the narcoleptic dog. So they studied the gene. Is there any mutation? So they came up with the finding that Orexin receptor 2 mutation in narcoleptic dog. So now you see that the difference. When come to mice, you get a narcoleptic symptom because in that one we are removing the orexin cells. When come to the dog, there is no loss of orexin cells. Then there is only mutation of orexin receptor 2. So now the scenario changes. So we UCLA group and the Stanford group, we have both group has the same finding. So when we looked, fortunately we had 3 human narcoleptic brain with the cataplexy. Okay, there are 2 types of narcolepsy. One, narcolepsy with the cataplexy. The other group, that is a very severe symptom. Then other one, there is no cataplexy. Cataplexy means you may have seen that people with a seizure, when in USA or in Europe, if the person with identified as a narcolepsy, they will not give a driving license because you cannot drive, because we don't know even a highway or anything. Anytime you can collapse, so it will they will not get a driving license. So we had human postmortem brain, and we looked into the hypothalamic area. What is the status of orexin in this human subjects? So we both simultaneously published. They published in Nature Medicine, and we published in Neuron. And we have only the pathological study, and they have the human subjects with the mutation and human patients, because they are we don't have the license for clinical studies. The Stanford group, it is most of them with the MD PhD. So these are the symptoms of narcolepsy, because it shows the excessive daytime sleepiness, cataplexy, sleep paralysis, and hypnagogic hallucination. So this is a sample of the human brain we used, and you can see that in the small box, this is the hypothalamus. So what we using that, we cut in, this is the Neurological and Stereology. This is a software computer, and it is integrated with the confocal or fluorescent microscopy. First, we prepare the slides. So it is a 40 micron thickness, and why we are using this stereology and neurological, it is for a qualitative and quantitative identification of the cells, and it makes our effort very smoothly, and it is you need only very small amount of time, because we have stereo. Now the analysis is becoming more complex, so we have to use a serial sections. So usually we will get a 1 in 500 micron or 1 in 1000 micron, and we prepare that section, and we use optical fractionator, and we have a very complex statistical program. It will allow us the estimation of the cells and the qualities and the properties and the morphology of the cells. So this is the first observation. In a normal human, there is a 70,000 orexin cells, and you can see that in the narcolepsy, there is almost 90% of the cells lost. So we also have some collaborative work with Stanford. It is a human narcoleptic patients, and collected the CSF, cerebrospinal fluid. So in the cataplexy patients, we cannot detect we cannot detect the hypocretin or orexin in the narcolepsy patients with the cataplexy, but there is no abnormalities with the hypocretin and narcolepsy without cataplexy. So this is just a comparative figure showing the anterior to posterior the distribution of hypocretin cells, and this is the dorsomedial hypothalamus, lateral hypothalamus, and posterior hypothalamus. And you can see that in the cell already very few cells you can see in the narcolepsy patients. So we also then looked into the projection area where the hypocretin fibers are going. So we almost looked where there is a high density of orexin receptor 1 and orexin receptor 2, and where there's a high projection areas of the fibers. And we can see that most of the fiber is projection area which is lost. So these all are the nucleuses in these are really hypothalamic nucleus, and there are some projection sites we quantified, and the entire narcoleptic patients with the cataplexy, the hypocretin system is entirely disappeared. So then we had to answer why and how. So one of the reason we are telling that there may be gliosis. Okay, we quantified the neuroglial cells, because if there is a neurodegeneration occurs in the brain area, the neuroglial cells will rapidly proliferate, and it is to counteract the compensation. So there is a internal damage in the brain, in the hypothalamus, where there is the neurons cell body is located. And we also looked into the fiber density areas, and we have seen almost 3 to 4 times the glial cells are elevated. So this is just comparing the anterior to posterior, the distribution. Okay, this is the hypocretin and MCH cells. And from the anterior hypothalamus to posterior hypothalamus, that means it is the near to midbrain. So there is a bell-shaped distribution, and the anterior part there is less neurons, and you can see that the mid hypothalamus, very high density of the neurons. And when this thing compared to control in narcolepsy with the cataplexy, we have not seen any abnormality with the melanin concentrating neurons. So you can see that very few cells in the narcolepsy with the cataplexy. So these neurons, both the neuron has role in a sleep-wake system, but MCH has in different stages. It is not associated with the lung. So this is just summarizing the differences between two narcoleptic pathology. So you can see that the narcolepsy without cataplexy, there is comparable cell present, and with the cataplexy there is a total absence, and in narcolepsy without cataplexy, we saw only in two areas, that is the posterior hypothalamus and lateral hypothalamus. There is a reduction. So now we are able to give some interpretation why there is a two types of narcolepsy. So one with the cataplexy, one there is no cataplexy signs. So that's a mild symptoms. So now you can see that how these symptoms causes the neuronal loss. We can see that, okay, there is very severe loss. Nearly 90% of the hypocrite orexin cells are lost. So when the cataplexy symptom occurs, it is basically the average occurrence of cataplexy in a narcoleptic patient is 18. So at the finishing of teenage, most of the narcoleptic patients has the cataplexy occur. So even though the first symptom at the age of 18, even we had the patients, most of the brain is the 70s and 80s. And we have some unpublished data. Even with the age of 18, the hypocretin CSF is undetectable. So that means any neurological diseases, Parkinson, Alzheimer's, anything, any type of neurological disease, the visible signs and symptoms occurs after maybe 1 decade. So in these patients, maybe the neurodegeneration started very early in the childhood. So we have seen many narcoleptic patients, and their life, they cannot do a normal life. And these patients has most of the neurodegenerative diseases symptoms. Not only the sleep problem, they have obesity, and they have low insulin resistance, diabetes, and they have early dementia, and they all have more or less mild neurological symptoms of other neurodegenerative diseases. So basically, the study is what we have done. Even in the follow-up of another category, narcolepsy become a model for other neurological diseases. So this is just a summary, graphic summary showing the normal, so the hypocortical distributions in the cell body. And you can see that with the cataplexy is almost 85 to 95%, and in the without cataplexy, this is only one nucleus in the posterior part of the hypothalamus. There is 35% of loss. So that is why the this narcoleptic patients has mild symptoms. So this is just summarizing the important area of the brain. So we added the hypothalamus, and you can see that the anterior hypothalamus, dorsal hypothalamus, posterior hypothalamus, these only 5 nucleus which is distributed the hypocretin cells. But when come to a cataplexy, this is just a summary of neuronal transmitters involved in the wakefulness and narcolepsy and cataplexy. So you can understand that how complex the situation and what are the structures are damaged. So any treatment, how we can treat this type of patients. And it is a very complex because the human symptom pathology we have not seen in any of the animal models. So in 2000, there is a different mice models, which makes our study more easier, and it is based on the hypocretin as the prominent neurotransmitter involved in this one, and the already abandoned narcoleptic dog. Even Stanford already closed out the Doberman narcoleptic dog colony, because there is no way it can compare the animal model and the human diseases. So then, because it is funded by NIH already told, okay, no more funding for dog study. So this is the summary of narcolepsy. So human narcolepsies have 85 to 90% reduction of hypocretin neurons. And surviving neurons have a normal morphology. And MCH neurons, which is intermingled with the hypocretin neurons, and there is no defect, no abnormality in with the MCH neurons. So when this, the 2000 finding, it's the NIH and the National Neurological Disorder Institute declared human narcolepsy as a neurodegenerative disease. So when we compared with this with the other neurological diseases in occurrence in American population, this will come fourth in category. It is a 1 in 2000. So this is the most specific in terms of cell loss. When compared with the other neurological diseases, there are multiple neurotransmitters which have abundantly damaged. So in case of narcolepsy, so far now, only one cell population which is solely responsible for narcolepsy with cataplexy. So now I am just moving to Parkinson disease. So how we move to Parkinson disease, to study Parkinson disease? So when we studied, we had only 3 human narcoleptic brain. And it is very difficult to get these brains. So we contacted in America and Europe. So it is extremely very difficult to get a human narcoleptic brain, because nobody is signing up, because they are not contributing the brain. So then we gradually explored, because Parkinson has more or less the same symptoms except cataplexy in narcoleptic symptoms. So we thought that there may be a link, there may be a role of orexin cells in a Parkinson disease. And it is a very easily available, the Parkinson brain, because there is a lot of study, because of all the concept is of Parkinson disease is based on dopamine cells in the substantia nigra, neuromelanin pigmented cells. So there are a lot more population and a lot more people with Parkinson symptoms. So we have a brain bank, and we have collected some of the brain. So this is just the before the orexin. This is the major pathology of narcolepsy, because you can see that when we cut the brain, slice the brain, we can macroscopically identify the pathology of Parkinson, because the midbrain substantia nigra, you can see that a black pigmented cells. That is the dopamine cells filled with the neuromelanin. So this is one sample of dopamine cells with the neuromelanin pigmentation. And this is a Parkinson patient brain cell. With you can see that the neuromelanin become aggregated. But in this one, you see that it has become dark. So that is the main pathology in a Parkinson patients. So basically it starts and all the dopamine cells which is filled with the neuromelanin pigmented cells. So even here, studying Parkinson in from human compared with the animal model, there is a big change because you can see in the animal model dopamine cells, but you cannot see these dopamine cells are filled with the neuromelanin pigmentation. So this is a basically sole property of human brain. All the dopamine cells in the substantia nigra, locus ceruleus, and ventral tegmental area, all are filled with the neuromelanin pigmented cells. Even in human, there are nearly 12 centers which has dopamine, even hypothalamus. And even you can give some of them with the olfactory bulb when nucleus accumbens. In that area, there is no neuromelanin pigmented cells. So the neuromelanin pigmented cells, when it starts in the human, usually by birth, when you are 1 year old, 2 year old, this melanin pigmented cells is not there. So it usually starts at an average age. It is with the two, the substantia nigra, locus ceruleus cells, ventral tegmental area cells. It starts getting neuromelanin pigmentation. So that may be one reason. Maybe here is telling that kids with a terrible tube because they are very hyperactive. So these cells mainly involved in the medulla and the locomotor movement activity. So Parkinson is a movement disorder. So now the thing is that even if you have animal models, we cannot 100% replicate the exact phenomena what in human. So we had collected Parkinson disease from our brain bank, and there are pathologically 7 stages and clinically 5 stages. Clinical stage 1, 2, 3, 4, and 5. At the stage 5, the person becomes vegetative. So in the stage 1, you will have a mild tremor or the movement a little bit shivering and everything. But when the progress, the stages are growing up, the stage 5, it is a very pathetic condition. So the person is getting vegetative. That means even your feeding also, you have to get a tubing. So luckily, we have all the stages Parkinson brain to study the same stereological and neurological quantification. So for that, we have a half brain, we collect a half brain. So we have to get a complete block of the brain, and we will get systematically cut, prepare the slide, and we do a random sampling estimation. So when we quantified the cells in our Parkinson, we saw the interesting thing that with the progress of the stages, there is a gradual loss, the number of hypocretin cells are coming down. So what usually the pathological staging is different. So whenever you see the clinical stage, that means the people usually the Parkinson occurs maybe after 60s. Now in America, it is coming early 50s. So by the time substantia nigra, the dopamine neuron, almost 80% is gone. That is why I told early with narcolepsy, for a clinical sign, for a neurological disease, it starts maybe a decade ago. So the people in the Parkinson field, they have a pathological chart. So the first symptom of Parkinson disease is to start with the olfactory bulb. So the first sign of Parkinson is that we have the problem with the smell. So that is the first sign. So even in the alpha-synuclein, the Lewy body deposit, that is the characteristics of Parkinson disease. So it initially begins with the olfactory bulb. There is even stage 1, you can see that there is not much loss in the hypocretin or anything, but your olfactory bulb, there is a dopamine neuronal population, and you will get a damaged cell population there. So usually the Parkinson identified by Lewy body alpha-synuclein deposit. So we also quantified what is the role of MCH. So this Orexin and MCH, they are just like the sibling. They are distributed in the same area and more or less same orientation. And we also see that there is a loss of MCH. So then in a narcolepsy, we don't have a MCH cell loss. So the hypothalamus of narcoleptic, Parkinson patient. So I have examined the hypothalamus of all category of Parkinson patients. So when we come to the later stages, when the brain, it is almost shrinks. And we had some Alzheimer's brain, and it is a very bad to see that type of brain, and it is already shrinking. And especially all the ventricular system, and it is exaggeratedly bulged. And many of the neural pathways, we can see that the thalamic tract, just like a wiring system, it has a bundles. So these all are damaged. So the MCH, if you look for other group, these all are a major damage, a structural damage with the disease. So this is the one group we are interested in the MCH because it is already have a role in sleep. So we also interested in this population. Even Parkinson patients will benefit because now the treatment in anywhere in the world, it is dependent on the dopamine system. That may be half-century old finding. And all the drug which is prescribing, dispensing, it is based on dopamine derivatives. So I have just a question for any neurological disease, any treatment which has a cure until now? No. Even the number of patients is increasing. Then where is the fault? Even what happened to the medical system? Even all the neurological diseases, day by day, increasing. and what are each country spending on the healthcare system and all the things, it is tripled. So what is the problem? Even though there are scientific community, researchers are giving advancement of the disease cause, the biotech companies are not interested. Even in 2000, we have discovered, even multiple labs across the world discovered the cause of narcolepsy. But in 2016, based on these findings, big pharmaceutical company introduced a drug that is not for narcolepsy. It is for insomnia. Why? Narcolepsy, you will not get the pharmaceutical company will not get any profit because there is really very limited people. If you say tell insomnia, 30-40% of the population, especially in developed countries, they are looking for anything like okay, even nutrient supplements, insomnia, or they will not have a good circadian or hygienic sleep, so they will go for a medication. So here, the medication, vitamin supplements, these all are all, everything is over the counter. Very few, most of them you will get over the counter, and people can consume as they want. But just in case of the case of the Parkinson disease, so there are so many other major findings contributed, but the medication, I have not seen any progress. Even a normal schedule for any drug come to from, we say that from bench to bed, that means from basic researchers to clinician. It is taking like 20 years. Even if it's a small company coming up with all the startups, or any small company coming up with this type of drug formulation, what they will do, that they will buy that small company, maybe double or triple the profit, and they will sell the company to the big pharmaceutical company. So this is an alpha-synuclein deposit. This is also a characteristic of Parkinson disease. So we just quantified what is the charge of alpha-synuclein distribution in the hypothalamus, where there is the hypocretin area. So we can see that based on the severity of the disease and the progression of the clinical stages, we can see that the alpha-synuclein deposit is going. So that means if there is an alpha-synuclein deposit, that means that cell is already malfunctioning. So we just double stained hypocretin with the alpha-synuclein, and we have not seen any alpha-synuclein deposit in the Orexin cells. But interestingly, there are a lot of cells with alpha-synuclein deposit. So there are other cells which is also damaged in the the Parkinson patients. So these cells are histamine cells. Histamine also has a major role in the wakefulness and sleep, and for the immunity. And histamine is the one most influencing the neuroglia cells. So this is just a summary of hypocretin and orexin and MCH pathology in all the stages. So you can see that stage 1, stage 2, stage 3, stage 4, stage 5, and this already gradually declining. That means most of the cells are gone. So we just quantified the area, the size. So there is the existing surviving cells don't have any abnormalities. So in summary, in Parkinson patients, there is a massive loss of orexin and MCH neurons. So how these neurons are related to the clinical stage or the duration. So these cells, in our analysis, it shows that the intensity of cell loss, both orexin and MCH, it is not the duration, it is the clinical stage. So in summary, not only the midbrain area, hypothalamic dysfunction has a major role in the clinical symptoms, and it has to be given more weightage to study the Parkinson patients. So now coming to another topic, how the orexin molecule is linked to opioid. So it is an interesting story. So when we studied Parkinson, we have a lot of Parkinson brain, but getting a control brain, that means without a neurological disease, it is very difficult. Even now, it is very difficult. So it is a long time wait. So when we requested more control brain, because the Parkinson patients are, we have to do an age matching control brain. So we got a brain more or less 63 years old. And when we studied that brain, and we have a surprising observation, it has a 50% of hypocretin cell increase. So it is very interesting. We are talking about hypocretin cell loss, and we got a brain which has a 50% of increased hypocretin cells. So we excluded that. That's an outlier. We excluded that brain, and we published the Parkinson study. Then we followed up the medical history of that patient. So when we got to detail, because when we get to the brain, we initially, they will give, because here is the HIPAA rule, the personal security problem, we will get only limited access to the database. So when we got more details about this subject, the interesting thing is that this person has 20 years of heroin addiction. So what is the novelty? So the triggering point is that the morphine can increase the orexin cells. So that is the initial observation we start. It is in 2008, and we don't have any opioid addiction brain. This is only one. So with a scientific study, anything with a n is equal to 1, we cannot do anything. So we need a significant number of subjects for a valuable statistical analysis. So we put a search in our National Brain Bank and European Union. We have a collection collaboration with the European Union and Netherlands Brain Bank Research. So they don't have anything with the hypothalamic area. So I started. The heroin is the basic molecule, is the morphine. So then, if we hypothesize that heroin causes the increase of hypoglycemia. So then the morphine is the base molecule of the heroin. Then we started doing animal experiments because we don't have any human brain other than this one, and we have no information about the animal models. So we started morphine injection into the mice. So because the morphine is an addiction field, and there are it is a very big field. You know, the addiction, even each type, nicotine, opioid, even opioid different category, even caffeine, and sugar. These all are separate pathways. So when I searched the literature, before injecting the morphine into the mice, we should have the pre-existing knowledge. That means we have to do the PubMed search and what is the existing status. So there are a lot of studies with the morphine, and some people also looked into Orexin relation, but there is no significant data available, and all the existing data is based on mild dose of morphine, and their duration is maybe 7 to 10 days. So we have planned different duration and a different dose, and we started the experiment. So in animal, we were able to replicate what we saw in one human brain with morphine injection into the mice. So that is the major milestone we overcome. So by the time it took from 2008 to 2013, we received 5 additional narcoleptic brain from US. Then there is a problem. These brains are teenagers. So below, these are teenagers overdose. So then we have to redesign, and we have to get additional control brain because it should age match. And that took also another 3 years to get sufficient number of human brain to complete that study. So this is a study we published in a science, one of the science publications, Science Translation Medicine. Opioid, the morphine, which increases the number of orexin cells in human and mouse, and reverses the cataplexy in mouse model of narcolepsy. So it is the interesting point is that we have both sides. In a disease, there are many diseases which the cells are losing, and this is the situation in opioid users. The number are increasing. So we designed in that way. We have to explain orexin has a major role in the opioid addiction. So the supporting factor in from the existing knowledge is that narcolepsy patients have no addiction. That is a promising. And we have the existing knowledge that one case report in 20 years back, one narcoleptic patient, he has kidney diseases and he has some transplantation, and he used morphine. So surprisingly, that patient has no cataplexy. So we connected these existing knowledge. So morphine can make a treatment for narcolepsy. And there is another thing is that there is a new neurotransmitter which is involved in the opioid addiction. So now everything is the existing knowledge is that opioid is a dopamine. So for this, we have did a substantial work, and this study involved 5 universities, and in 3 countries. It is involved in from the Japan one lab, and from Netherlands, and John Hopkins. So this is the human data we have with the 5 subjects. So in all the subjects, we saw that a uniform pattern of hypocretin cell increase. So you can see that there is the opioid addiction, how with the 1 or 2 usage of these addictive compounds, how these people are becoming addicted. So there is a major change with overdose in our brain. So this is the one focus we did. So it has a structural change. That is why all the addict, even alcoholic, nicotine, they are very demanding for this substance. Why? Because their internal structure, the internal built-up, it already, there is a drastic change in the nervous system. So this is the dose response in animals we have done. So we have done different parameters, fixed dose, escalating dose, and from 1 milligram to 100 milligram, and we have different duration. So 14 days and 60 days. So even there is an optimum dose which has the major influence in increase the number. So that is the 50 milligram. So you can see that after 50 milligram, there is no increase. It is gradually when you increase the dose, it is coming down. So even a long duration, it is not giving in that much increase. Even it also, there is a tendency to damage the system. That means the cells may be die. So we just quantified in hypothalamus which area, even though we say that the orexin cell population, and it has each nuclei has a specific characteristics. So it is not all the cells has the same property. It has a different afferent and efferent connections. So for the morphine, these cells affected differently. So basically the lateral hypothalamic area, which has the most influence in the increase of hypocretin cells. So why the lateral hypothalamic area? This is more close to the thalamic tract. So that is the outgoing from the hypothalamus, it is the outgoing wiring system. So we saw that it has the lateral hypothalamic orexin cells. It has the major influence in the opioid addiction. So then we also looked into what is the withdrawal when we stop giving morphine, what happens. So we also quantified the morphological changes. So there is a the effect lasts up to 4 weeks. The significant effect only up to 2 weeks. So up to 4 weeks, these cells, the size is coming to back, and the cells, the elevated population is coming down. It is coming to back to the normal level. So this is one mice neurological plot giving an example. So then we also looked into the precursor proteins, and there are many other proteins co-localized with the hypocretin cells. So we saw interesting cell that the prepro-hypocretin or prepro-orexin, that is the precursor of the orexin cells, from there it generated. So it also the precursor level, the mRNA is already increasing. It is not only the orexin, it's also the precursor also increasing. That is very interesting. So then we also checked some of the co-localized neurotransmitter like neuronal pendaxin and dynorphin, and these all the cells, these neurotransmitters also increased. So interestingly, we looked into the melanin concentrating hormone, and it is the neighbor of the hypocretin cells, and there is no significant change. So this is just for showing we are validating the orexin specifically have major role in the opioid addiction. So now there's a question. We have to answer these new neurons, how it is developed. So we looked into some of the neurogenesis. So new cells is developed, or some other neurons are converted. So we just made it clear that there is no neurogenesis. So we did something, a special marker for neurogenesis. It is the BRDU and the double coating. And in the hypocretin area, we have seen no neurogenesis. But in our adult mice, the dentate gyrus, there is always the new neurons are forming. So we have got some staining, some cells in the dentate gyrus, but in the hypocretin area, there is no cells. And this is the figure 1, this is the A, which shows that this is the wild type. And this is we also confirmed any changes, it is specific to orexin neurons. We have a orexin knockout mice. So we used that also to confirm orexin is needed. In an orexin knockout mice, there is no availability of orexin. So there is nothing happened with the orexin knockout mice. And now we thought that, okay, it is a good idea and it has a clinical implication. If this can be used, deployed as a treatment for narcolepsy. So for this, we collaborated with an investigator in Japan, and he developed a narcoleptic model more resembling human narcolepsy. So we collaborated with that lab, and in that model, that mice model, the advantage is that you can deplete the orexin cells how much percentage you need. So if you want to deplete 100%, You can 100%. So we depleted 30% of the cells. Then why 100, not 100%? If we deplete 100%, we already know that hypocritic knockout mice, there is no effect of morphine. So we given morphine to these animals, and we repeated the same experiment what we did with the knockout, not knockout, the wild type mice. And we saw surprisingly in these hypocritin depleted mice, it is coming back to the normal. So then we also looked into the cataplexy symptoms. Then from the Netherlands group, they have a big brain collections pathology. And we got some narcoleptic with the cataplexy, and they have in their treatment received morphine. So we evaluated that one. In that one, we saw that increase of the loss, even though the normal with the cataplexy there is a 90% loss, but even with the morphine treated, there is a tendency to increase the number. So that is an interesting thing, and morphine can be used as a treatment of narcolepsy. But the problem is that morphine we cannot use as a treatment for narcolepsy because morphine is a controlled substance and it induces addiction. We saw that this opioids, the malpractice of the healthcare system and the people, it is almost opioid national emergency. So the clinicians, they never support morphine to treat for the narcolepsy. Now, what is the solution? So now there is a substitute for morphine, and they taken out the characteristics of morphine. It causes addiction. So that is a synthetic drug under pipeline. In that case, there will be no addiction. So I already told Orexin and other neurological diseases. So these are some examples: narcolepsy, Alzheimer's disease, multiple sclerosis, traumatic brain injury, Huntington disease, Parkinson disease, Niemann-Pick disease, multiple system atrophy, depression. So these are the existing and some supporting literature, and they already have some quantified data for Orexin. So now you already saw that with the same point, the same neurotransmitter, there is a 90% loss, and there is increase. So how this is happening? And we also demonstrate that there is no neurogenesis. Then how this is happening in our human brain? So why morphine brings the increased number of cells? So opioids, the action site is opioid receptors. And this is the most abundantly distributed receptors in our brain. And this is not only in our neurons, it is in our glial cells. And this is also involved, this receptor is also involved in the pain management. So most of the nervous system, even in the brain, spinal cord, anywhere, this is a very complex. So in the hypocretin orexin cells, almost 70% of the cell population has the most important opioid receptor, mu receptor. So we propose that when morphine enter into the body, the orexin precursor or the hypocretin precursor, the prohypocretin, it may be bind to the inactive form. The bound form means it is attached to a receptor. So when morphine comes, it is a high affinity molecule. So then they release the preproxin. So what happened to the preproxin? It will convert to hypocretin. That is the simplest mechanism we are explaining for the addiction. Then what is the, so in that way you can use, or you can either develop a treatment for the addiction. So then what about narcolepsy? So narcolepsy, it is that there is an inhibitor. It may be different sources. So now there is a group from Switzerland coming up that it is the neuroglia that it has a major role in the death of hypocretin cells. So when we answer one question, and there is hundreds of questions arising, so how deep the nervous system? So these are the collaborators, and we from UCLA, and I also have appointment with the Veterans Medical Center in Los Angeles, and we have collaboration with John Hopkins, and we have collaboration with the Netherlands Institute, and from the Netherlands, the University Leiden, and we have Japan. This is from Japan we got the animal model for the opioid study. So thank you very much for listening to the talk.
Thank you, sir. It was an informative presentation. So now we move on to the discussion part. Your queries can be posted in the comment box. Additionally, anybody who likes to ask questions directly can unmute yourself and ask the questions.
Hello.
Hello.
Can you hear me?
Yes, yes.
So initially in the introductory part, you were talking about the neuroglial cells that control the neurons.
Yeah.
Okay. What is the role of the astrocytes and the ependymal cells?
So basically, now there is a big importance and focusing on neuroglia cells,
Yes.
which has broadly classified into 3 groups.
Okay.
So astrocytes,
Uh-huh.
microglia,
Yeah.
and oligodendrocytes.
And what about the ependymal cells?
Ependymal cells, it is already distributed to the wall of ventricles.
Yeah. Yeah.
So that
Okay.
is
Okay.
for the
CSF.
yeah.
Okay.
So why the neuroglia cells are important means because there is a lot of studies showing that the
Hmm.
glial cells also equally important in the maintenance of the homeostatic mechanism of the brain cells,
Or the neurons.
neurons. So
Okay. Okay.
just we saw that okay when you put a just histamine is basically inflammatory response,
Okay.
and if there is an elevated histamine,
Hmm.
you can see that the neighboring the in the same location
Hmm.
there is maybe 3 times more the glial cells responding.
Glial cells responding to that in that specific area.
Yeah, a specific area.
Okay. Okay. So then you are talking about the pre-pro orexin,
Orexin. Yeah.
which is two type, which is a two types. That is
Preprorexin is the
orexin 1 and orexin 2.
Orexin 1 and Orexin 2 that it is derived from the preprorexin.
So it is actually produced as a precursor.
Yeah, it is a precursor. The
It is
precursor
a
for it
single.
is the
Is
preprorexin.
is it a single polynucleotide, which is polypeptide, which is cleaved into two specific
Yeah, two.
moieties,
Yeah.
which has got two different receptors?
Both has affinity for receptor 1 and 2.
Both yes.
Yeah.
And the
And
receptor and yes. Continue, please. Continue, please.
basically we focused on we already studied the orexin B. There may be a natural question that what happened to the orexin
Uh-huh.
B. So
Right.
basically we studied it is both are the same cells.
Okay. 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 So
So
that is
it has the
the
affinity of orexin receptor 1 and receptor 2. And
and it
the
will
distribution of the receptors are not from some location you can see orexin 1, then orexin 2. Then
okay.
most of the area you can see both receptors.
Okay. Whether it's a extracellular or intracellular, extracellular receptor.
It is extracellular.
And the receptor will act as first messenger or second messenger, first messenger or second messenger concept.
It's a first messenger.
First messenger. So it will bind. It will bind.
Yeah.
And bring about the cyclic AMP reaction inside the cell.
Yeah.
Okay. And then you are talking about the gene mutation.
Okay.
You are talking about gene 2. I can't understand what is meant by that gene 2.
It is the study by Stanford group.
Uh-huh.
And
So what is it? Why they call it as gene 2 and which chromosome it is located?
It is the receptor 2 gene.
Okay. Receptor 2 gene, in which chromosome it is located?
It is 17, chromosome 17.
Chromosome number 17.
Yeah.
Then
So
you were
Stanford group is more they are the clinician group.
yeah.
And MD PhD, and basically they are focusing on human. They have a wide collection of human narcolepsy. It is a US National Narcoleptic Center.
Okay.
And
So
they had a narcoleptic dog.
Mm-hmm.
So
but that you were telling that it has no connection with the human beings.
yeah, because our study shows that there is no even they already came up with that there is no mutation in the human subjects.
Okay. Then we are talking about the hypocretin cells.
Yes.
And you are just saying that is 70,000. Is this the total number of cells or is the number of located in a particular area?
It's exclusively located in the hypothalamic area. And we do quantification of the complete hypothalamus. and it is an estimated population is 70,000.
Okay, the total number of cells.
Yeah,
And what is the quantum of the?
including both
Then
half.
okay, then what is the quantity of the orexin secreted by them?
Which one?
What is the quantity of the orexin? What can be the quantity of the orexin that can
It's
be secreted?
quantified in a normal hypocretin. It is and hypocretin CSF. It is something in between 400 and 500 milligrams.
Milligrams. Okay, okay.
Micrograms. And nanograms.
So is it nanogram or picogram?
It's a picogram.
Yeah. Sorry, because I am really surprised to hear that if it is only 70,000 cells and
it is not a milli. It is a picogram.
it is picogram. Because I have gone through this topic now. Just to make, I was reading some of the topics. So then you are talking about the neurogenesis.
Yes.
Said what is meant by BRDO and double?
It is a neuronal marker, and it
okay. Yes.
When we stain that one, if it is a newly formed cells, only newly formed divided cells will stay.
BRDO means what?
It's something bromo. I have to check.
That is bromophenol blue method.
Yeah.
The bromophenol blue method is actually the staining of proteins.
Yeah, it's the new cells with the protein.
Okay, and what is in the double casting?
Double coating also it's for the new neurons. It is only exclusively only in the new neurons.
Double coating.
Yeah.
You are coating it with antibody or stain?
Yeah, antibody.
Okay.
It is the immunostaining.
Oh, okay. It is immunostaining. Okay, right. And then about the PD.
Which one?
About Parkinson's disease.
Yeah.
So you mean that the Parkinson's disease, there is the secretion of dopamine, and the dopamine production, once it is going down, is it the dopamine getting degenerated and forming a cast, a rounded black cast?
Yeah, it is the major cause, the degeneration of the dopamine system.
Yeah.
And in a human, the substantia nigra, the midbrain area, and the locus ceruleus, there is the dopamine cells are filled with the neuromelanin pigmented cells.
Okay, that is the MCH is having a role there.
MCH is in the hypothalamus.
Okay.
It is not it is not related to dopamine.
Okay. So thank you very much. You have done. I was just keenly hearing the whole session now. It was really informative, and I wish you all the best. You go ahead, and you'll definitely go to the heights. Thank you.
Oh, thank you.
I would like to interrupt. Please, participants, please note that the feedback link for the feedback form is provided in the comment box. It is also available in the comment box in the YouTube live streaming mode also. Again, sir, we have a question in the comment box by Akash Kannan. Has any study done using orexin receptor agonist?
Yeah, there is a lot of study with the orexin receptor antagonist. So it is for the narcolepsy, and even now they are deploying. We have not done, but there are literature available even for the opioid addiction. Orexin, then orexin receptor antagonist, and there are a lot of studies are available.
Any more questions?
Sir, the session was very informative. I'd like to ask you, in narcoleptic patients, can the
Yes.
changes in orexin levels interfere with the dopaminergic pathways and cause any issues with the structures in basal ganglia and extrapyramidal structures in the later stages of the disease?
Yes. Hello.
Yes, sir.
So what is the question?
In narcoleptic patients, in the later stages of disease, if the orexin levels are constantly changing, can it interfere with the dopamine pathways and cause problems with the basal ganglia or extrapyramidal structures?
So in a narcolepsy, with the cataplexy, the first sign occurs on average age is 18. So, in the clinical study, we find no detectable hypoketosis. So there is how it is totally absent. It is undetectable. The hypoketosis of hypoketin in that one, it is undetectable. So then there will be not much or the CSO and the cyclical function fluctuation. There will not be much there. Basically, it is totally absent. So how the orexin, the lack of orexin causes Parkinson means Parkinson patients, because it has it is lacking most of the stimulatory factors. So it is directly related with the sleep pattern.
Okay.
Any more questions? I hope there are no further questions.
Okay.
If there are no further questions, we can proceed to the last part of the session. I would like to remind the participants to fill the feedback form before leaving the meeting. Now we have the vote of thanks. I invite Dr. Jisha Jacob, coordinator, for delivering the vote of thanks.
Good morning. With immense pleasure, I would like to thank today's speaker, Dr. Thomas Chacko, for his informative talk on importance of hormones in neurological activities. He had explained the role of orexin in sleep and stress in a promising way. Dr. Thomas is a person with immense experience in the field of brain research and lot of publications in international peer-reviewed journals with a very high impact factor. So we are extremely happy to have you as a resource person today. Thank you, sir. Thank you very much for your time and support. I would like to express my sincere thanks to the college management and Principal Dr. Sabuke Thomas for their wholehearted support. I also express my sincere gratitude to Head of the Department, Dr. Bobby Jose, and my colleagues for their enthusiasm and support. I am happy that many faculty members from various colleges across Kerala participated in today's webinar. I thank them for their valuable time and cooperation. I thank all the participants, especially researchers and students, for their kind cooperation. I hope you all enjoyed the program. Thank you. Now, we conclude the webinar here. Before leaving, I request you to fill in the feedback forms from the link available in the comment box to get the e-certificate. Thank you.